Hello guys, welcome back to my lessons. So let's answer questions under the topic of DNA code of life. So the first set of questions says that table 1 shows DNA based triplets that code for different amino acids. So if we look at table 1, they've given us the amino acids. As I always say, you don't have to know those amino acids by heart. You'll be given a table with amino acids, and then on the other side they might choose to give you DNA triplets, or they might give you tRNA or mRNA. So here we are given base triplets of DNA template. We have GGG, AAA, CCT, TAT, CGT, TTA, TTA, right? So table 2 shows a part of the sequence of anticodons that code for a polypeptide. Table 2, we have amino acid 1, 2, 3, 4. We have anticodons, UUA, CCU, AUA, UUA, right? So they say identify using the tables above amino acid 2 and 4. So what are you going to do? You must look at anticodon 2, which is CCU, and anticodon 4, UUA, and find the corresponding base triplets of those nitrogenous bases, and then look for it on the table there on DNA, and then you will look at their amino acids. So as I always say, that you're going to write DNA, under DNA you write mRNA, under mRNA you write tRNA. So you are given a nitrogenous base CCU for tRNA. You're going to write it on the tRNA CCU. So because now we are looking for DNA, why are we looking for DNA? Because DNA triplets, they're the ones which are going to help us to find amino acids, right? You move from DNA to mRNA. To mRNA, C pairs with G, C pairs with G, U pairs with A. Now let's find the DNA. G pairs with C, G pairs with C, A pairs with T. So CCT, let's look for it on the table. The first table, we have CCT, the amino acid is glycine. So let's look at the second nitrogenous bases. We have antigone UUA at 4. And then we're going to find mRNA first. So this sequence helps you. You don't just jump from tRNA to DNA. You move from mRNA first, tRNA first to mRNA, and from mRNA to DNA. UUA, U pairs with A, U pairs with A pairs with U. Now let's go to DNA. A pairs with T, A pairs with T, U pairs with A. So we have TTA. Let's look for TTA on the table. It's alanine. So it means the answer there is 2 glycine, 4 alanine. So let's look at the next question. They say how many amino acids will be coded for by the anticodons at table 2. So let's look at let's count the amino acids there at table 2. We have 1, 2, 3, 4. So your answer is 4 amino acids. So the next question says name and describe the process that uses anticodons to make proteins. In this topic, we've spoken about DNA replication, we've spoken about transcription and translation only. So if you are looking for processes, choose from the 3. So the process that only include anticodons, the tRNA, it includes also proteins. That is translation, not transcription. So let's describe it. You write translation, you're going to get a mark for it. Each tRNA carries a specific amino acid. That is the first point. When the anticodon on the tRNA matches the codon on the mRNA, the tRNA brings the required amino acid to the ribosome. Amino acids become attached to each other by peptide bonds to form the required protein. That's all you're going to get your 5 marks. So let's look at the next set of questions. It says the diagram below shows the process of protein synthesis You can see transcription there, you can see translation. We have a DNA molecule, we have an mRNA molecule, we have a tRNA there. So let's look at the questions. Identify molecule X, that is a DNA molecule. Organelle Y, that is a ribosome. Identify the nitrogenous base labeled 1, 2. Labeled 1 and 3, right? Nitrogenous bases. So how do you find them? We have an mRNA there which has the nitrogenous base AUCCAAGA, and you're lucky because they've written them for you there, AUCCAAGA there on the ribosome. And then as you can see that 1 pairs with C there, 2 pairs with C, and then number 3 pairs with A. So you're moving from mRNA to tRNA. You go to the sequence again, DNA under DNA, mRNA, and mRNA, tRNA, and then you write for mRNA because that's what you're given. You are given C, C, A. Going down, C pairs with G, C pairs with G, A pairs with U. So your 1 there is G, which is guanine, and then your 3 there is U, which is uracil. So describe the role of DNA during transcription. DNA codes for a particular protein but cannot leave the nucleus. One strand of DNA is used as a template to form mRNA. That is symbol. As you can see that these lines were taken from transcription. So let's look at the next question. It says describe the part of protein synthesis shown as process W, which occurs at organelle Y. That is translation, it happens in the cytoplasm. So according to the codons on the mRNA, tRNA molecules with matching antigons bring the required amino acids to the ribosome. This is called translation. The amino acids become attached by peptide bonds to form required protein. Or you can write it this way. Each tRNA carries a specific amino acid. When the antigon on the tRNA matches the codon on the mRNA, then the tRNA brings the required amino acid to the ribosome. Amino acids become attached to each other by peptide bonds to form a required protein. So let's look at the next question. The table below shows the amino acids that correspond with different DNA codes. We have amino acids and then we have DNA codes on the side. They say write down the correct sequence of the amino acids copied for by structure S. Let's look at structure S is the mRNA in the diagram on the previous page. So they want the correct sequence of amino acids, right, using this DNA codes. So we are given DNA, we are given TCT, we are given TAC, we are given GGT, right? Let's go to mRNA. T pairs with A, C pairs with G, T pairs with A. For this base triplets, T pairs with A pairs with U, C pairs with G, and then G pairs with C, G pairs with C, T pairs with A. So we have the mRNA. We have the mRNA, and then let's look at the let's look for the tRNA as well. So we have the A pairs with U, G pairs with C, A pairs with U, A pairs with U, U pairs with A, G pairs with C, C pairs with G, C pairs with G, A pairs with U. So you need to know which amino acid comes first, which amino acid comes the second, and which one comes the third, right? TCA matches with which base triplets on the mRNA. It matches with AGA. Let's look at AGA. It's the last one there, so it means it's going to be the last one. Arginine is going to be the last one. And then let's look at TAC. TAC matches with AUG. AUG is the first base triplet, so glycine will come first. And let's look at GGT. GGT matches with CCA. Where is CCA? CCA is there in the middle. It means methionine will come in the middle. So the sequence is going to be glycine first, and then methionine second, and then the last one is going to be arginine. So let's look at the last set of questions. It says that the diagram below represents the process of protein synthesis in a cell. As we can see, this is a cell. Those are the nucleus. We have the DNA molecule. We have mRNA. We have the tRNA there. Name the process which occurs at W, that is transcription. At Y, that is translation. Organelle X is the nucleus. Molecule Z is an mRNA. Another question says, state two locations of DNA in a cell other than in the nucleus. It is found in chloroplast, in a plant cell, and in mitochondrion also. So let's look at the next question. Says, describe the process at W. The process at W, that is transcription. The double helix unwinds and it unzips to form two separate strands. One strand is used as a template to form mRNA. Using free nucleotides from the nucleoplasm, the mRNA is complementary to the DNA. mRNA now has the coded message for the protein protein synthesis. Let's look at the next question. The table below shows some tRNA anticodons with their corresponding amino acids. I told you they will give you a table of amino acids, and then they'll either give you DNA, mRNA, or the tRNA base triplets. So in this case, we are given tRNA. Name the DNA base triplets that codes for serine. Where is serine? Serine is there. We are given the tRNA, which is the UCG. So UCG for tRNA, we're using the sequence. Let's substitute UCG, and then let's go up because we are looking for DNA based triplets. We go to mRNA. U pairs with A, C pairs with G, G pairs with C. Let's go to DNA. A pairs with T, G pairs with C, C pairs with G. So the answer is TCG. So let's look at the next question. It says that the first two amino acids are coded for by the molecule Z in the diagram. Let's look at molecule Z. And the first two amino acids coded. So the first two amino acids. It means that from Z we are going to take the first base triplets, the first codons, which is UAC and GUA. So those are on the mRNA, and then now we are looking for the tRNA because it is the one on the table. Let's go down. U pairs with A pairs with U, C pairs with G, G pairs with C, U pairs with A pairs with U. Where can you let's look for AUG. AUG, it is tyrosine, that is the first amino acid. And then CAU, let's look for CAU, that is valine, that is the second amino acid. So the answer is tyrosine and valine. Those are the two amino acids that they want. What is the change in the sequence of nitrogenous bases in DNA molecule called? That is gene mutation. That is a change in the sequence of nitrogenous bases. So the last question says the codon as CUU, last codon on the molecule Z changed to CCU, right? It changed to CCU. Explain the effect that it would have on this particular protein molecule. So this is simple. What you're going to tell them is the anticodon will be GGA instead of GAA, and then the last amino acid will be proline instead of leucine, resulting in a different protein. Thank you so much for listening. Don't forget to subscribe to the channel, and I would really appreciate it. Thank you.